Hello class, my name is Mrs. Peltier, and today we're going to learn about DNA. DNA is the base of all of us. It's what makes you and you. It gives Charlie his red hair, Scarlet her blue eyes, and so on. DNA has to be copied to make new cells. This is a strand of DNA. It's kind of like a train track. It's long and winding. The body wants to make two different tracks because there is more demand for the railway. We have 4 workers that will help build the new railway. We have helicase, or Helen, that will unzip and break apart the old track. We have primase, or Perry, who will make a new track to show where the track should go. Then we have DNA polymerase, who is the DNA builder that will build the new track. And lastly, we have ligase, or Larry, who will glue the track together. Here again we have our old railroad track, and our friend Helen is going to break it apart. So now that Helen has broken apart our railroad track, we only have half of each track that's finished. So it's Perry's turn. Perry is going to make railroad tracks that go left and right. So he's going to start on the right side of the top, and he's going to build a little track that is going to show the DNA builder where to go. Next, we have the DNA builder that will build right to left on the top strand until it gets to the fork in the road. And again, Perry's going to lay down a piece of track to show the builder where to go. And now the builder has to go left to right, so they build across left to right. Now again, because Perry can only build left to right on this strand, he's going to start on the left side, and the builder is going to build the rest of the track. Next, Larry is going to glue the track all together so it doesn't break apart. Now we have our new track, and I'm just going to erase it. Okay everybody, thanks for watching. I hope you learned something about DNA copying. Have a good day. Bye. Good morning everybody. In this class, let me just erase the homework from last day. This class, like we learned last class, is going to be on DNA. Does everybody remember DNA? Well, today we're going to work on DNA replication. Exciting. So everybody remember that DNA is the building blocks of life, and we got to replicate it once in a while so our cells can regenerate. So here's a strand of DNA, and we're going to go ahead and copy it. So the first thing we're going to do is we're going to break apart our DNA. So that's done by helicase, which is an enzyme that will unzip the DNA strand into 2 separate strands, creating a separation fork, which is that Y shape you've got. So the top strand from right to left is going to be 5' to 3', and on the bottom it's 3' to 5'. Remember, we know this because DNA is anti-parallel. That means the complementary strands run opposite from each other, like we learned in the previous class. So on the top strand, we're going to build from 5' to 3'. So we lay down RNA primer, called primase, and it's going to be the starting point for our DNA. Then DNA polymerase is going to build continuously from 5' to 3', right to left. But on the bottom strand, primase is going to start like around the middle because it has to build from left to right, 5' to 3'. So DNA polymerase is going to build left to right, 5' to 3', and then primase is going to start again to the left, and DNA polymerase is going to build the rest. So again, the top strand is called the leading strand. We call it the leading strand because it has no trouble building from right to left, 5' to 3', so it'll just build continuously. The second one is called the lagging strand because it has that trouble. It can't continuously build because it has to build towards the replication fork. So on the leading strand, the primer is laid down, and basically the DNA polymerase just builds continuously down the line. No problem. And on the lagging strand, because it can only be made in the opposite direction, it is made in fragments. These fragments, or chunks, are called Okazaki fragments. RNA primer is put down, then the DNA polymerase puts down a short fragment from 5' to 3' direction. And then this process is repeated. The primer is laid down, and then the Okazaki fragments are laid down from 5' to 3'. Once the new DNA is made, the enzyme exonuclease knocks out all the primers, and the DNA polymerase fills in all the gaps. Then finally, the enzyme ligase seals up all the DNA fragments, so they're glued together. So DNA replication is called semi-conservative because it's made up of one parent strand, which is in blue, and a newly synthesized strand or daughter strand that's in green. Alright guys, thanks so much for watching, and I hope to see you next class. Hello class, today we're going to be talking about DNA again, like the previous class. In the previous class we talked about DNA structure. Today we're going to talk about DNA replication. There are two major steps in DNA replication. First, separation of DNA from its strands. And secondly, there's the building of complementary strands. Here we have a strand of DNA. The strand is going to be unwound by the enzyme helicase, and gyrase is going to relieve any tension that occurs. The enzyme helicase is used to break hydrogen bonds that hold complementary parent strands together, creating an unzipping of the helix. This terminates at the replication fork. Then, binding proteins anneal to the template strands so they do not re-anneal. Then the enzyme primase lays down RNA primers where new complementary strands will be made. DNA polymerase III starts to add deoxyribose nucleoside triphosphates from 5' to 3' of the new strand. DNA polymerase only builds from 5' end to 3' end of the strand. One of the strands is called the leading strand. It is made continuously towards the replication fork. The other strand is called the lagging strand. It cannot be made continuously because it runs in the opposite direction, because DNA is anti-parallel. Remember that anti-parallel means the complementary strands run opposite from each other. RNA primer is put down on the lagging strand, then the DNA polymerase puts down a short fragment from 5' to 3' direction. Because the lagging strand can only be made in the opposite direction, it is made in fragments. These fragments, or chunks, are called Okazaki fragments. As you can see here, the Okazaki fragments on the bottom strand are in green. This process is then repeated over and over until it gets to the replication fork. Once a new DNA is made, the enzyme exonuclease knocks out all the primers and the DNA polymerase fills in all the gaps. Finally, the enzyme ligase seals up all the DNA fragments so they don't fall apart. DNA replication is called semi-conservative because it is made up of one parent strand and one newly synthesized strand, or daughter strand. Thanks everybody for watching my new YouTube video on DNA replication and the simple steps to the not so simple steps. Your homework is on the board and it's due next class. I'll be checking it, so you better do it. Thanks so much for watching. Bye guys, love you.